Welcome back, students. This is Module 2.2.6, Enzymes. Now, enzymes are a special class of biological macromolecules. The vast majority, 99.999% of them, are proteins. Most of them are globular proteins. And we will consider pretty much that all of them are proteins for our purposes at this time in this class. The definition of an enzyme is an enzyme is a biological catalyst. Now that doesn't help you very much if you don't know what a catalyst is. So a catalyst is a chemical that speeds a reaction but is not itself changed during that reaction. And therefore, when it's over, the catalyst can perform the same function again. It can catalyze the same reaction again. And so biological catalyst is simply a catalyst inside a cell or inside a living organism. As I said, all enzymes are pretty much globular proteins. Very few are RNAs. Now, the thing about enzymes is that they have a very particular three-dimensional shape. And this is critically important because this is what creates the high specificity or high selectivity of enzymes for a single substrate. When we say substrate, we simply mean a molecule that the enzyme can catalyze in a reaction. Let me, just to introduce enzymes, give you 5 facts about enzymes that will help you to better understand them. First, enzymes don't make anything happen that would not happen anyway, but they increase the rate hundreds to millions of times faster than in the absence of the enzyme. This can be critically important when, for example, a biological function is being catalyzed. Think if you've ever held your breath underwater. It's not good enough to say that you will eventually get a breath of air. You need that breath of air when you need it. And so this is one of the things that enzymes allow to happen, is reactions to be catalyzed in a timely manner, allowing life to occur. A second enzyme fact is that the enzyme is not changed in the reaction, and it can be used over and over again. A third enzyme fact is that the same enzyme usually works for both the forward and backward reactions. If, for example, we have a reaction A plus B produces C, then we can have C breaking down to form A and B. In both reactions, the forward and backward reaction are catalyzed by the same enzyme. A fourth enzyme fact is that each enzyme is very selective about the molecules upon which it works. And you should know why at this point, with knowing what you know about protein structure. And I'll give you more examples of this further along in this lecture, but I expect you to be able to explain why enzymes have high specificity, why they're highly selective about which molecules they work upon. The fifth and final enzyme fact that I'm going to share with you is that extreme conditions, high temperatures, harsh conditions, extreme pH, extreme osmotic environments where they're very salty, can cause an enzyme to denature, to lose its shape, and when it loses its shape, it loses its function. In order to comprehend enzymes better, we want to ask a basic question, and that is, why do chemical reactions occur in the first place? And we can answer this and say that a reaction occurs when the participating molecules collide with some minimum amount of energy known as the energy of activation, or the activation energy. And enzymes function by lowering the activation energy, and they do this by holding and positioning the reactants in an optimal way. They may strain chemical bonds that need to be broken. They might create concentrations favorable to the reaction, but they allow reactions to happen at a much faster rate. Here's kind of a whimsical representation of enzyme function. Well, I guess this isn't the whimsical one. My mistake. This shows a very important reaction. This is called a rate diagram, and along the horizontal axis, this is time, or progress of the reaction, and the y-axis, or the vertical axis, has energy levels. And so we can see that we start out here where I'm indicating with the cursor that this is the energy level of the reactants. And we can see that the activation energy without the enzyme is a pretty steep obstacle that has to be overcome. And then the larger molecule can be broken down to smaller molecules with lower energy levels. And what is the difference in energy? Where has that energy gone? Because energy is certainly conserved, and that is was released when these chemical bonds were broken. So energy is released in this reaction. We can see a second pathway of the same reaction with an enzyme, and the energy of activation shown by the green portion of the curve is considerably lowered, and this happens through the optimal holding and positioning that enzymes are able to perform. And I think you should be able to draw something like this, showing a reaction with and without and the energy levels. Here is my whimsical picture. It's showing at what rate princes are produced from frogs, and this wall is the energy of activation. And here the wall is much smaller, and so we can see that the reaction occurs much smaller. Much more rapidly. So we've talked about why chemical reactions occur at all, but now we want to talk about different ways to speed up a reaction. So here are 4 ways to speed up a reaction. One of them is heat, and heat puts energy in, and it causes the participating molecules to move with greater speed, increasing the number of collisions, producing more productive collisions, if you will. A second way to speed up a reaction is to increase the concentration of the reactants. And once again, this makes more collisions, and more of those collisions are productive. A third way to speed up reaction is to decrease the products. Now, if you think about this, if the reactants and products are in the same system, every time you have a collision between a product that has already reacted and a reactant that hasn't reacted, this cannot be productive. And so by decreasing the concentration of the products, it's kind of another way of increasing the concentration of the reactant, so more of the collisions are in fact productive collisions. And the fourth and final way to speed up a reaction that we're considering here is with catalysts. And life of these 4 different ways has chosen to use catalysts. If we increase heat, and they can be very problematic for living things, for cells that are made of and contain many proteins, which can denature at high heat. Increasing the reactants can mess up the needed control of metabolic pathways, and the same with decreasing the products. And so catalyst is the mechanism that life settled upon to use to control or to speed up reactions. And so enzymes, biological catalysts, allow reactions to occur at low temperatures without changing the concentrations of reactants and products. So now let's look at 2 different models of enzyme function. The first model is called the lock and key model, and over here we can see this little animation of a key. And the thing about a lock and a key, although it has high specificity as enzymes do, a lock and key is also very rigid, and the rigidity of this lock and key is not quite accurate. At least that's the way that biochemists do not think it's very accurate. So this is the old model. A more recent expression of an enzyme model is a hand and glove, and that's also known as AKA induced fit. And if you think about the way a hand goes in a glove, when it goes in, it changes the shape, and so it is that enzymes and substrates change one another. And so the hand and glove or induced fit model describes the changeable interactive aspects of enzyme substrate interaction. So this is a more updated model. Here's a diagram of an enzyme, and it shows the active site with the substrate in it, the little pink molecules there. This is crazy because I'm sorry, that was not the substrate. The substrate is right up here, the red. But it's crazy because it shows how large the enzyme is relative to the substrate. And this is just a schematic model of an enzyme. And I have more of that to show you here, kind of to give you the whole rundown in a schematic way of how an enzyme works. So if we look here, we can see the enzyme looks like a little purple boat with a very specific shape that will fit this molecule. This molecule is sucrose, which consists of fructose and glucose bonded together, and they fit exactly inside the active site of the enzyme, and this forms something called an enzyme substrate complex, and within the complex the bond between the two is broken, between the two monomers is broken, freeing up glucose and fructose, and the products are released, and the enzyme is able to go and do it again. And so this shows over and over how an enzyme can function to catalyze a reaction. It's very important to understand that enzymes mediate pretty much all of the metabolic pathways inside cells. It's kind of hard to envision that. These don't occur in necessarily specific structures. Some of them certainly do, but some of them occur, some of the metabolic reactions occur in the cytoplasm. And the enzymes are simply there or not there. And so the issue of control is something to consider. And so we'll just consider it briefly here, enzyme control. We're going to consider one mechanism of enzyme control. There are several others. The method we're going to consider is feedback inhibition. And I want you to remember that metabolism consists of many biochemical pathways, all of which are under the control of enzymes, as I was stating earlier. So let's look at feedback inhibition. In feedback inhibition, a final end product serves as an inhibitor of the biochemical pathway that produces it. So that is what feedback inhibition is. This is showing a series of chemical reactions, in fact 5 chemical reactions. Each has an enzyme that mediates it, enzyme 1, enzyme 2, enzyme 3, enzyme 4, enzyme 5. And the end product in this case is tryptophan. Tryptophan is one of the 20 amino acids. We didn't memorize the names or anything, but you might recognize tryptophan as that amino acid that makes you sleepy when you eat turkey. It's also in milk. Warm milk releases tryptophan. In any case, in this pathway to produce tryptophan, tryptophan itself serves as an inhibitor, and it will attach to the active site of enzyme 1 in its pathway. And so when you get a high concentration of tryptophan, it will bind to the many copies of enzyme 1, shutting down or at least slowing down the metabolic pathway. And it's not a complete on and off, because it's unlikely that every single enzyme 1 is going to have a tryptophan attached to it, but the majority will, and this will slow production to a very low level. And then when tryptophan becomes rare in the cell, when it's used up and it is needed, a mechanism exists to release the tryptophan from the enzyme and to reopen this pathway and resume higher levels of production of tryptophan. This is going to conclude our discussion of enzymes, as well as the topic of cellular transport, before we've slipped enzymes in. So we will resume later. Thank you for your attention.